Everyone knows that animals either lay eggs or give birth, but what most may not realize is that these two options are far from the only ones. On top of that, how did such a thing evolve? How can we know? One bizarre little tadpole-shaped fossil from the badlands of Montana provides some clues as to how bebes were being born 300 million years ago. In the animal world, you've got two major ways to pop them kids out. One is birthing. You got that jelly bean growing all up in the wet parts, and then it comes out all squirmy like. The second way is popping out a bunch of eggs. The eggs are just an external baby-making oven that gestates the jelly bean a bit longer so the mama can go about her business. So, just like with most subjects, that is the easy part. It's the simple biology of the matter. So of course there's a lot more to it in the real world, and that's why you're here. Not to be content with the basic bio you learned in 5th grade, but to actually progress and grow as a person. So, in reality there are, give or take, about 5 modes of reproduction. There's oviparity. Fertilization is external, the oocytes being released into the environment and fertilized outside the body by the male. This is common in mollusks, arthropods, and fish, and is found in most frogs. Then there's oviparity. Fertilization is internal, but the female lays zygotes as eggs with a substantial quantity of yolk to feed the embryo while it remains in the egg. The egg is not retained in the body or only for a limited time. Oviparity is found in insects and birds. Among mammals, the monotremes are oviparous. Next up is ovoviviparity. Oviparity with retention of zygotes in either the female's or in the male's body. But there are no trophic interactions between zygote and parents. This mode is found in the slow worm Anguis fragilis. In the seahorse, zygotes are retained in the male's ventral marsupium. In the frog Rhinoderma darwinii, the zygotes develop in the vocal sac. In the recently extinct frogs Rheobatrachus, zygotes developed in the stomach. Then there's hysterotrophic viviparity. The zygotes develop in the female's oviducts but find their nutrients from other tissues, whether skin or glandular tissue, oophagy or adelophagy, intrauterine cannibalism in some sharks, or in the black salamander Salamandra atra. Finally, there's hemotrophic viviparity. Nutrients are provided by the female, often through a placenta, as in mammals, except for marsupials and monotremes. In the frog Gastrotheca ovifera, embryos are fed by the mother through specialized gills. The lizard Pseudomoya paganstecheri and most mammals use this form of viviparity. For those interested, there's also placental viviparity, arguably the most highly developed form of viviparity. Placental mammals, including humans, are the best known example, but adaptations in some other animals also have incorporated this principle or close analogies. Other examples include some species of scorpions and cockroaches, certain genera of sharks and snakes, and velvet worms. In general, viviparity and matrotrophy are believed to have evolved from an ancestral condition of oviparity and lechithotrophy, nutrients supplied through the yolk. One traditional hypothesis concerning the sequence of evolutionary steps leading to viviparity is a linear model. According to such a model, provided that fertilization was internal, the egg might have been retained for progressively longer periods in the reproductive tract of the mother. Through continued generations of egg retention, viviparous lechithotrophy may have gradually developed. In other words, the entire development of the embryo, though still with nutrients provided by the yolk, occurred inside the mother's reproductive tract, after which she would give birth to the young as they hatched. The next evolutionary development would be incipient matrotrophy, in which yolk supplies are gradually reduced and are supplemented with nutrients from the mother's reproductive tract. In many ways, depending on the ecology and life strategy of the species, viviparity may be more strenuous and more physically and energetically taxing on the mother than oviparity. However, its numerous evolutionary origins imply that in some scenarios, there must be worthwhile benefits to viviparous modes of reproduction. Selective pressures have led to its convergent evolution more than 150 times among the vertebrates alone. There is no one mode of reproduction that is universally superior in selective terms, but in many circumstances, viviparity of various forms offers good protection from parasites and predators and permits flexibility in dealing with problems of reliability and economy in adverse circumstances. Variations on the theme in biology are enormous, ranging from trophic eggs to resorption of partly developed embryos in hard times, or when they are too numerous for the mother to bring to term. But among the most profoundly advantageous features of viviparity are various forms of physiological support and protection of the embryo, such as thermoregulation and osmoregulation. Since the developing offspring remains within the mother's body, she becomes, in essence, a walking incubator, protecting the developing young from excessive heat, cold, drought, or flood. This offers powerful options for dealing with excessive changes in climate or when migration events expose populations to unfavorable temperatures or humidity. In squamate reptiles in particular, there is a correlation between high altitudes or latitudes, colder climates, and the frequency of viviparity. The idea that the tendency to favor egg retention selectively under cooler conditions arises from the thermoregulatory benefits and that it consequently promotes the evolution of viviparity as an adaptation is known as the cold climate hypothesis. Viviparity has been extensively documented among the various clades and grades of fish. Just so everyone is clear, not everything that is called a fish is strictly evolutionarily derived from a common ancestor. Since technically all life is derived from a common ancestor, everything is related to everything else. However, with regard to fish, there are a few groups that group with other groups of animals more than they group with each other. For example, the lobe-finned fish, the Sarcopterygians, are our cousins. We share a common ancestor with them. The ray-finned fish, the Actinopterygians, do share a common ancestor with us and the lobe-finned fish. The cartilaginous fish, the Chondrichthyans, split off the jawed fish group a long time before the bony fish, making them much older with a history that saw adaptations to all sorts of things. Viviparity is quite common among the fish, with examples from the sharks and rays, the elasmobranchii, the teleosts, modern coelacanthiforms, and even the Urodel and Sicilian amphibians. But all known, modern members of the Chondrichthian group, known as the holocephali, are reported to be oviparous, making them very odd exceptions. The Baric Ult Limestone is a continuously consistent layer of rock that all dates to a specific time, with specific fossils in it that covers a large area and is sandwiched above and below between other layers of rock from before and after it. The Baric Ult Limestone is more specifically slapped with the label of lens because the layer is shaped like a lens, fat in the middle and skinny near the edges. And since the general processes of the rock cycle are a thing, no one formation is ever never ending, so the Baric Ult is free to pinch off the extra bits to be a fun bean shape. The Baric Ult Limestone is found in Montana and has been dated to 324 million years ago. At this chunk of time, the Chesterian stage of the Mississippian subperiod of the Carboniferous period, the region was covered in mudflats, floodplains, and lagoons, filled with both brackish and fresh water. So why is this formation seen as a logosteton? That boils down to a few hypotheses, with a general agreement that the critters that died in the water in the area were able to survive the processes of decay long enough to be quickly buried and preserved nearly perfectly. So this could mean that there was an anoxic or oxygenless layer of the various lagoons. Anything that stayed there too long would die and fall to the bottom, or anything that died above the layer would subsequently sink below the layer. The layer would keep the vast majority of scavengers and bacteria away from the bodies, halting the decay process long enough for fine features to be preserved. Another possible explanation, in part or in whole, is that huge, periodic mudslides would swing on into the lagoons after heavy rainfall and violent storms to cover and kill anything that wasn't fast enough to get out of the way. Some evidence against this being the main vector of perfect fossilization is that many fish are found with their gills distended from their bodies, which is what is usually seen in asphyxiation. The world in which Falcatus lived was chock full of all sorts of strange fish. Paleontologist and Bear Gulch expert Richard Lund published a paper in 1980 in the journal Science in which he described 2 fetal specimens of a chondrichthyon from this Bear Gulch rock layer that show a very interesting example of viviparity in the cartilage having weirdos. The 2 specimens, UM6148 and CMNH35455, are distinguished by their distinctive cranial anatomy, tooth plate numbers, body form, and incipient cranial ornamentation. They are both tadpole-shaped fetuses of large size, specialized toothers, and lack of developed fins, showing that they are unrelated to any of the other 5 holocephalans in the Bear Gulch assemblage with similar features. The holotype specimen, UM6148, this little guy right here, is 35 millimeters, 1.4 inches long, preserved outstretched on its side with a slight bulging little belly. Outlines of calcified cartilage on the head indicate a short snout, large dorsal ventrally unrestricted orbits, and a mouth of relatively short gape and holostylic suspension as in the Chimeriformes. Holostylic jaw suspension is found in most elasmobranchs and all bony fishes. The upper jaw, known anatomically as the palatoquadrate, is loosely attached by some ligaments and cartilages to the skull. The only thing that braces the jaws against the skull is the hyoid arch, all of the little bones that make up the tongue and throat bones, like the hyomandibular, ceratohyal, and basihyal. The second specimen, specimen CMNH35455, this other little guy right here, is 29 millimeters, 1.14 inches long, fully scaled and preserved on its side with a big, full, protruding tummy covered in even more scales, somewhat resembling a yolk sac. The eye and gill arch pigments are preserved, as is a bolus of material at the rear of the abdominal region that strongly resembles the amorphous calcium phosphate typical of coprolites. Little guy had a tummy so full of soup that he tuckered himself out and died on the spot. Both specimens have the same toothy tools in their tiny jaws. They have a few plates of teeth in their upper jaws. The frontmost plates are the smallest, with both the front and back ones having a series of sub-equal sized cusps. The back plates are much more like compressed blades with coarse serrations. The lower jaw had quite different tooth plates, with much taller, farther spaced apart, recurved, tall, and thin cusps filling out a lot of space in their little mouths. As Richard Lund notes, the dentition, as a whole, forms a sharp-edged tearing and piercing structure totally without precedence among recent or fossil holocephalans. Keep in mind that the holocephali is the group to which modern ratfishes and chimeras belong, but was far more diverse in the past. The bulgy-wulgies, bloated tadpole-like body shape, general curvature, and no fins indicates that these creatures are fetuses, but do not give any indication of the size they were at birth. The tummies full of soup are internal and cannot be a yolk sac, as shown by the complete scaling and lack of an umbilicus. On top of all that good stuff, the large poopy blob in the smaller specimen implies a considerable amount of feeding. Little guy was chomping. The greatly expanded foregut of the modern intrauterine feeding shark Lamna is essentially the same as in these fetuses. Modern intrauterine feeding sharks and amphibians absorb the relatively limited amount of yolk at an early stage. Subsequent growth of the fetus is supported by oophagy, or eating of other eggs, or, in the more complex amphibians, by uterine secretions. Most modern viviparous sharks are supported fetally by trophonemic structures. In 2 families, nutrition is supplied through yolk sac placentation. Modern holocephalans just ingest their own yolk. Large, well-developed slashing and piercing dentition is unusual in a fetus, and even more unusual in a holocephalan. Modern holocephalans are oviparous, and there are no known tooth differences between adults and young. The teeth of modern and known fossil holocephalans varies from plucking and shearing plates to durophagus plates made for crushing hard things. While the teeth of these fetuses could be interpreted as that of a predator, the teardrop-shaped body and fetal condition all but prohibit a predatory lifestyle. Lund therefore interpreted the teeth as specialized for opening egg capsules during intrauterine feeding. Functioning dentition in intrauterine feeders among both sharks and amphibians has been reported to differ from that of the adults. So, good old Lund decided to name these little varmints Delphyodontos dacryformes, from the Greek delphos meaning womb, odontos meaning tooth, and dacryform meaning teardrop-shaped. There are 5 other Bear Gulch holocephalans with similar body forms, only one of which has been described. The minimum body size of well-developed, apparently free-living young among these species is about 16 millimeters. The maximum body size of sexually mature adult males is 150 millimeters. The fetal Delphyodontos is unusually large for members of its taxonomic group in the assemblage, and the cranial calcifications indicate an unusually high fetus-to-adult size ratio, characteristic of oophagus or egg-eating forms. In view of the well-developed fetal dentition of Delphyodontos, the evidence of feeding in utero, the abdominal distension strongly suggestive of a specialized foregut for oophagy, and the unusually large fetal size, it must be concluded that Delphyodontos is viviparous. Thus, it constitutes the first direct evidence of viviparity in the holocephali and in the Paleozoic chondrichthyans. Biologist John P. Worms discussed factors considered significant in the evolution of viviparity in sharks. The implication of the first phylogenetic position is that holocephalans, being more primitive, are oviparous. According to Richard Lund, neither part of the statement is acceptable. Worms could find no correlation between viviparity in recent sharks and their geographic distribution or by habitat depth. Finally, Worms justifiably dismissed purported osmoregulatory considerations. Stuff to do with how fish regulate their molecules and ions and stuff. Little is known about the food habits or feeding ecology of sharks as they age, except that the young are frequently found in different places than the more migratory adults. The suggestion that egg-eating produces a predator trained before birth is not acceptable for modern holocephalans, among which no known members can be considered predaceous. However, there is a strong correlation of viviparity with large adult size, which in turn has implications for feeding ecology. Viviparous species produce not only larger offspring, but also fewer of them. Large size is an absolute advantage in reducing the number of potential predators. Fewness in the number of young is an absolute advantage in reducing competition for food among them. Large size and low numbers may have been a particularly advantageous combination in a community such as Bear Gulch, where the extreme diversity of the chondrichthyans indicates very fine-grained partitioning of resources based in part on size and age segregation. There were too many cartilage cretins, so they all needed to be eating specific and different things in order to avoid as much competition with one another as possible. Chondrichthyan viviparity has evolved independently many times and must be seen as of considerable selective advantage by virtue of, rather than in spite of, the production of fewer larger young. The probable evolution of viviparity with dental specializations for intrauterine feeding in a Paleozoic holocephalan reinforces the suggestion that this is evolutionarily the simplest method of viviparity, requiring no specialized maternal structures and few fetal modifications. Unfortunately, no adult forms of Delphyodontos have ever been found. It's very possible that these are a weird fetal form of any number of known holocephalan members of the Bear Gulch, but it would be impossible to confirm this without a mother's body full of bloated little womb-toothed gremlins. Therefore, the adult may either be a slightly more mature-looking version of the tadpole beast from hell with bigger, nastier teeth and a more streamlined body, or the adult may have been a more normal holocephalan. However, the term normal could never apply to this group of cartilaginous fish, even to this day, as they are all the weirdest, freakiest little ocean squishies to ever bear a spine. Delphyodontos lived alongside many strange creatures, like the chondrichthyans, Agassizetus, Lystracanthus, and Falcatus. It also lived alongside many ray-finned fish, like Discocera and Paratheraceus. Other fish included the rhabdodermatid Cardiosuctor, the rhizodont Strepsidus, and Hardistiella, one of the oldest known lampreys. The invertebrates of Bear Gulch were truly diverse creatures, like the hoplocarids, which include modern-day mantis shrimp, Andorella, which is the youngest known Synzyphosaurine, and more enigmatic creatures like Typhlosis, and these square objects which might be sea salps or cnidarians. Other invertebrates include mollusks like the nautiloid Tylonautilus, pteroid bivalves which have been found encrusting sargassum like brown algae, as well as productid brachiopods, Paleolimulus, phylocarids, and echinoderms like crinoids, echinoids, sea stars, brittle stars, and a many-armed starfish called Lepidasterella montanensis. Not much more can be said of Delphyodontos. It's even possible that more specimens of these little sea demons exist out in the Bear Gulch, or even in collections yet to be described. Only time will reveal more secrets of the bloated little womb tooth. For more interesting stories about nature, the history of life, or what goes bump in the night, subscribe, like this video, drop a comment in the comment section below, and hit the bell icon to stay in the know with everything Edge. Thanks for watching.